We're going to be talking about our manuscript published in this first issue of Cancer Discovery by Peter Hammerman, Martin Sauce, Alex Ramos, and colleagues, and on the discovery of DDR2 mutations. Excuse me. First, quickly want to mention conflicts of interest, as is done in AACR slides, and point out that this does not include Bristol Myers Squibb, the manufacturer of dasatinib, which I will be discussing. Squamous cell lung cancer is one of the major forms of lung cancer. As Dr. Wong-Ki Hong mentioned this morning, the overall incidence of lung cancer leads to the deaths of more than 150,000 people in the United States each year, and more than 1 million people worldwide. By comparison, more than die from colon, breast, and prostate cancer combined, indicating the seriousness of the disease. Squamous cell lung cancers account for nearly 30% of these lung cancer deaths. In recent years, as Dr. Garraway has just discussed, we've made major advances in lung adenocarcinoma treatment. He was referring to the treatment for ALK fusion lung cancer, now treatable by crizotinib, and also for the treatment of EGFR mutant lung cancers by gefitinib and erlotinib, an area in which the discussant, Dr. William Pao, has made enormous contributions. But there are very few known targets for treatment of squamous cell lung cancer, and there are no approved targeted therapies to this date. In a systematic screen in my group, we analyzed all protein tyrosine kinase genes for somatic, that is to say cancer-specific mutations, in paired tumor and normal DNA from lung squamous cell cancer patients, sequencing the DNA both from the cancer and from normal lung tissue. The result we obtained was that mutations were found in several kinase genes, including the DDR2 gene, where mutations were found in 11 out of 290 lung squamous cell cancer cases, or a total of between 3 and 4%. DDR2 encodes the discoidin domain receptor 2 tyrosine kinase, which was not previously known to be recurrently mutated in cancer, I should say. A few scattered mutations had been reported. And the stick figure at the bottom demonstrates the locations of the mutations throughout the predicted protein sequence, including mutations in the discoidin domain and a number of mutations, including several clustered ones inside the protein kinase domain. And I'm going to come back to this 765 to 774 region slightly later. We asked whether there were any existing protein kinase inhibitors that potentially could inhibit the growth of squamous cell lung cancers bearing DDR2 mutations. We had identified 2 squamous cell lung cancer cell lines that harbor DDR2 mutations and that require DDR2 for their survival, as shown by RNA interference experiments. Dasatinib, of all the drugs tested, was the most potent at preventing growth of these cell lines. Dasatinib is a multi-kinase inhibitor that inhibits both the ABL and SRC kinases, and it's approved for treatment of chronic myeloid leukemia, which is caused by the BCR-ABL fusion. And DDR2 mutant lung cancers, squamous cell lung cancer cell lines shown here are killed by dasatinib. So first of all, a PDGFRA mutant lung cancer cell line known to be inhibited by dasatinib, shown here in blue, is a positive control for killing. A KRAS mutant cell line, the mutants that, types of mutants we've just heard about from Dr. Chinayan, except the point mutants, is resistant to dasatinib, and the 2 DDR2 mutant cell lines are both highly sensitive. We went on to test these dasatinib in animal xenografts of these cell lines and found again that it could prevent tumor formation in the animal models. Our colleagues, Eric Hora, who's a co-author, had been a leader of a clinical trial of a combination treatment of dasatinib with the EGFR inhibitor erlotinib, and in that trial, 1 patient showed a partial radiographic response to the combination treatment. This patient's tumor did not harbor an EGFR mutation, which would explain a response to erlotinib. However, work by Martin Sauce and Roman Thomas showed that this tumor did harbor a mutation in DDR2, S768R, predicted to activate the DDR2 kinase. And so shown here is just CT scans demonstrating the response. 2 months before erlotinib and dasatinib combination treatment, you can see a large mass here in the left lung because the orientation of CT scans is reversed, and within 2 months of treatment, significant decrease in the size of this mass. So the mutation found here was S768R, and I mentioned that's actually the mutation that's modeled in the image that's on the cover of the first issue of Cancer Discovery. And I mentioned that there were several other mutations in this domain between residue 765 and 774. So finally, what are the implications for the diagnosis and treatment of squamous cell lung cancer? First, the role of DDR2 in the causation of squamous cell lung cancer needs to be further validated, including demonstration causation in animal models. Second, given that dasatinib is already approved for therapeutic use, it is worth exploring clinical trials of dasatinib for squamous cell lung cancer. Third, it is also worth the development of further monospecific and multispecific DDR2 inhibitors. And fourth, squamous cell lung cancer diagnosis should include analysis of candidate drug targets, including EGFR, FGFR1, as shown recently by Weiss et al. In Science Translational Medicine, and PIK3CA, the PI3 kinase catalytic subunit, as well as DDR2, arguing for a pan-targeted approach to analyzing squamous cell lung cancers and the development of targeted therapies. Finally, I want to thank the leaders of this discovery effort. Peter Hammerman is a medical oncologist working in my group at Dana Farber. Peter did most of the cellular studies, including the RNAi experiments and the demonstration that dasatinib is effective against small cell lung cancer cell lines. Martin Sauce is a postdoctoral fellow working in Roman Thomas's lab, who analyzed a large set of the mutations described and demonstrated the mutation in the patient of Eric Horace who had responded to dasatinib. So I thank you very much for your attention, and I will turn it now to Dr. Baselga.
Thank you so much, Matthew. And we would like now to invite Dr. William Pao, who is the Director of Personalized Cancer Medicine at the Vanderbilt Ingram Cancer Center, to discuss this paper. William.
Well, I too want to congratulate the editors as well as the authors for launching such a successful first journal for cancer discovery. To be an honor to be part of this panel. So I'm very excited about the paper by Dr. Meyerson and colleagues because essentially we have a new target for therapy in squamous cell carcinoma of the lung. And as Dr. Meyerson alluded to, lung cancer is the leading cause of cancer-related death in the United States and worldwide. This very clearly depicts on the right with statistics from the American Cancer Society that lung cancer is the leading cause of cancer-related death in both men and women, with over almost 200,000 cases a year and 150,000 deaths a year. So this is the way that we were taught to think about lung cancer in medical school, and this is probably the way we've been thinking about it for more than 100 years or so. Basically, what do the lung cancers look like under the microscope? This is the general distribution. The two major types of lung cancer include small cell lung cancer and non-small cell lung cancer. And then non-small cell lung cancer has three main types, including adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. And actually, up until maybe the past few years, this information was enough to help guide therapy in terms of what we would give to our patients with metastatic disease. Essentially, we needed to know if a patient had small cell lung cancer, and they would get one type of treatment, and then if they had non-small cell lung cancer, they would get one type of treatment. However, what's really become evident through a lot of molecular analysis, cancer research, and cancer genome efforts is that non-small cell lung cancer is actually very heterogeneous and comprised of multiple different subsets of cancers that can be defined by what we call driver mutations or mutations in signaling proteins that not only make the tumors grow, but then are required for tumor maintenance, such that if you can target the signaling protein that's mutated, you can actually get a very effective anti-tumor effect. And so, as I said, here is the traditional view of lung cancer. Dr. Chinayan and Garraway talked about KRAS mutations. That was actually found in a lung cancer patient in 1987. We know it's in a large fraction of non-small cell lung cancers. In 2004, multiple groups found the EGF receptor mutations, and then already by 2009, we could start to define non-small cell lung cancer by these different driver mutations, including those driven by EGF receptor, a mutant ALK, mutant HER2, and so on. And most importantly, many of these are now amenable to specific targeted therapies, notably the mutant EGFR can be targeted with EGF receptor tyrosine kinase inhibitors, and mutant ALK can be targeted by ALK tyrosine kinase inhibitors. And there's more information on this at the website or published. So what about, what I didn't mention actually is, unfortunately, most of these driver mutations have been found in adenocarcinoma as opposed to the other subtypes of non-small cell lung cancer. And so squamous cell carcinoma has been a little bit considered to be the odd man out, not only because we haven't found a lot of targets there, but also because a lot of the therapies we have now exclude patients with squamous cell carcinoma. So as I mentioned, most of the drivers mutations that we found are found in adenocarcinoma. There's a drug called Avastin or bevacizumab, which blocks the growth of new blood vessels. That's approved for use in non-small cell lung cancer, but in people without squamous cell carcinoma. There's another drug called Alimta that can be very effective, but that also is approved for use in patients without squamous cell carcinoma. And there's actually no drugs that we have that are specifically indicated for squamous cell carcinoma. So sometimes in the clinic, when you tell a patient they have squamous cell carcinoma, you know, their face drops because there's fewer options in terms of treatment. And so this is why I'm very excited about Dr. Meyerson's paper. This adds to the now growing list of new targets that are being identified in squamous cell carcinoma. He mentioned FGFR amplification, which was just reported in December of last year by Dr. Thomas's group. This looks very promising as a target, and FGFR tyrosine kinase inhibitors are in development. There are a number of other mutations, and now we can add DDR2 mutations to that. It may be a small fraction, but notably we already have drugs that are available to target this specific mutation, and so hopefully we can now begin to have specific therapies for patients with squamous cell carcinoma. Thank you.